Hello, everybody, and welcome to this live stream from Dementia Researcher. My name is Adam Smith, and I'm delighted to be your host today to explore an exciting funding opportunity that's making a real difference in accelerating progress in dementia research. I'm going to bring in my wonderful guests today. Today, we're turning a spotlight on Race Against Dementia's Ignition Fund, a unique and flexible seed funding that helps researchers act quickly on unexpected findings and new ideas. Whether it's a breakthrough in the lab or the result of news that sparks a new hypothesis, this fund exists to fuel early discovery and rapid follow-up. And as I mentioned, I'm joined by three brilliant guests, some of whom you will have met before through our podcasts or live streams. I'm delighted to welcome, first of all, Molly Huggins from Race Against Dementia, who's a research lead there, who will talk more about the vision behind the Ignition Fund and how it works. Hi, Molly.
Hiya.
And then we also have Dr. David Koss and Dr. Aitana Sogorb Esteve, who are two researchers who were funded by this program last year, and they're going to talk about their experiences in applying for the fund and offer tips for future applicants as well, and talk a little bit about their research so you get a sense as to the kind of research that this fund has funded in the past. But before we begin, let me just show you the program again. So there we go. First of all, we're going to hear from Molly, who's going to give you some background information about the grant application process and about what they're looking for. And then we're current projects from Aitana and Dave, who are going to talk about their work. And then we'll have a chance as well at the end for you to put your own questions to Aitana, Dave, and Molly, and ask us anything you need to know. You can do that at any time during the live stream. If you just post your questions, if you're watching on YouTube, if you post those in the comments. If you're watching on X, I think if you click the recording, there's a chance to add those alongside, and again in the comments on LinkedIn or Facebook or wherever you happen to be watching this. And if you're watching this on recorded, we'll give you an email address a little bit later on where you can do that. But wait until you've heard from Molly first, because hopefully your question might already be answered in Molly's talk, and that's probably enough from me.
Everyone, I'm Molly, and I'm the research officer at Race Against Dementia. So thought I would begin just by telling you a little bit about the charity and what we do. And I know you've heard a little bit from that video about how we were founded. We were founded by three-time Formula One world champion racing driver Sir Jackie Stewart. When Sir Jackie was a racing driver, Formula One was a very different sport, and it was a lot more dangerous than it is today. It wasn't uncommon to have really tragic fatalities on the track. I think back then the statistics were, if you were driving for 5 years, there was about a 2 in 3 chance of you dying on track. So it's a lot different to the sport today, and that's really down to Sir Jackie's tireless campaigning. When he retired from driving, he decided to put all of his efforts into campaigning for better safety, from introducing full face helmets, proper seat belts and trackside barriers, which today seem like no-brainers, but back then they were faced with a lot of contention. But thankfully, due to all of his campaigning, those changes have now been made. And then fast forward a bit further along into his retirement, and his family was faced with a really life-changing news that his wife, Helen, was diagnosed with dementia, with frontotemporal dementia. And so Jackie devoted all of his time and his efforts into trying to find the best treatments and the best possible care for Helen. Unfortunately, he was faced with the reality that there is no cure for dementia, and there were no treatments that could really significantly slow the progress of Helen's dementia. And so that's when he decided to set up Race Against Dementia. And we exist to raise funds to identify, fund and guide pioneering early career researchers from all across the world. And we use the pace and precision of Formula One to do so. And it is really tragic that these two industries have come together under this kind of circumstances. But what Sir Jackie has really identified is a way to bring together an industry that he really really familiar with, the world of Formula One, with its technology and its pace, and bring it together with dementia research. And what we want to do is to approach dementia research differently and ultimately speed up the progress of research. And so the inspiration behind the Ignition Fund really comes from the way that Formula One teams build their cars and their success. So in Formula One, a team doesn't just build one part of the car before moving on to the next. They run everything in parallel because time really matters. In contrast, scientific research is often quite constrained by a linear funding model where you apply for one grant and then halfway through you need to be thinking about the next one, applying for the next one, and so on. And that's where the Ignition Fund really comes in. So it's a precision injection of up to £100,000 for you to fund a new line of inquiry alongside your existing research. So it's really a chance for you to fund a parallel project that can build on the existing work that you're already doing. And it's really designed to be fast and agile so that you can get access to that funding and really quickly and keep up your funding momentum. And so all UK-based early to mid-career researchers are eligible to apply for this grant. You can apply for up to £100,000 and the maximum duration of your project can be 24 months. We really want to see applications based around looking into or working towards preventions and treatments for dementia, and we welcome applications across all different kinds of dementia as well. So what we're really looking for is for researchers who have a new lead, a new signal, something your current funding structure can't immediately support, but that could really excel your progress if acted on now. Because we know that just like in motorsport research, the pathway to a discovery or breakthrough is very rarely linear, and new leads can emerge very unexpectedly, or you can have new ideas midway through a project. And without flexible funding resources, those ideas really risk being lost or worse, completely forgotten about and never acted upon. And so this is a really nice quote from Sir Jackie, and the Ignition Fund is really what we call the F1 way of doing science, because in Formula One, if there's a problem with a car, it's fixed by the next race because they can't afford to wait. So they have to really push boundaries of engineering in real time, and that's really what we look for in Ignition Fund applicants, is someone who has that urgency and the real want to move forward faster. And now we know £100,000 can't cure dementia, but what we do expect it to do is to enable you to push forward an idea or a project a lot faster than you could without it. And so the application process is designed to be quick and simple, and there are just 2 stages. So you have the application form and the interview. So the application form can be found on our website, and it's designed to be quite similar to your standard grant application, where you need to explain what it is that your project is, how you're going to go about it, and why it really matters. But there are 2 questions that we asked that I just wanted to highlight because they're really important in this application. So the first one is why this is an unexpected element of research. So you need to explain the finding that you produce that was surprising, unexpected, or goes against the accepted school of thought. And it's really your chance to show your innovation and the novelty of your idea. And the second question that we ask is, how will this project create new and exciting findings from your current work? So you really need to show how this project could lead to exciting discoveries based on the work that you're already doing. So tell us what your current work is and how this grant would enable you to pursue a novel idea or finding that your current funding couldn't support straight away. The second part of the application form is the lay summary. And this is really important because it's assessed by a panel of people living with dementia or people who care for people living with dementia. And they'll look at how your research could impact the real lives of people with dementia. So in your lay summary, we need to avoid using super technical language and try and keep it quite plain but very informative, and use compassionate language as well. So make it really clear why your project matters and how it's going to help people with dementia in the future. And so if your application is shortlisted, you'll be invited to an interview. And the interviews are quite short. You'll be asked to give a brief presentation about your project, and then you'll have a chance to respond to any questions or concerns that the panel might have surrounding your application. And after the interviews, the panel will discuss, and then they'll make recommendations for funding. These recommendations will be presented to our board of trustees, and then we'll have final approval. So as I said, this application form can be found on our website, and the deadline to get that back to us is the 12th of September at 4 p.m. And all you need to do is fill out the application and then email it to ignitionfunds@raceagainstdementia.com. And the interviews will be on the 25th of November this year, and they'll be virtual. So here are just a couple of points to think about when you're writing your application that our panel members really look for. So the first is the unexpected element. Just to reinstate again, you really need to show a new lead or a surprising finding that has emerged from your current work that could lead to something a lot bigger. And the second point there being the novelty. So this fund and this charity really is built for boldness. So we're really interested in original perspectives and fresh thinking. So make sure that you're really showing that in your application. And again, that parallel extension, we really want to see a clearly defined parallel direction that could be made possible with this support. So we want to understand how this extends your current research. And then finally, the final point there being succinct and clear, which I'm sure goes without saying, but we really want to keep it tight and concise for the application form. And actually in each section, it has character limits, because we really want clear and focused answers to make sure that you're keeping to those character limits as well. And then finally, before I hand over to Dave, some common questions that we get asked. So the first is if you require a detailed project costing report. We do ask you to there's a budget section in the application form, but it doesn't need to be super detailed. Something brief is fine, like a table and a brief justification, but we don't need anything super detailed. And I should also note that the funding isn't applicable to lab space or overheads, and we really do intend the funding to be for people or consumables. And the second question there is if you require letters of support from head of department or collaborators, and the short answer is no. The only documentation that we need from you to apply is the application form, and that's the only document that the panel will use to assess your application. And finally, do you need to include figure or image in your application? You can, and there is a space in the application form where you can upload just one figure or an image, and you're only allowed up to one per application form. It's not a requirement, but it is optional, and if you feel like it will support your application, then absolutely please do. So all this information is on our website, raceagainstdementia.com, and if you have any further questions after today, you can email us at ignitionfund@raceagainstdementia.com, and someone will get back to you. And this is just a final picture here of, you can see Aitana there, of our lovely network of researchers. So anyone who is funded by us will join this really global network, and we do get together quite often. This is all of them outside Aston Martin F1 factory. So that is all from me, and thank you very much for listening. I'm very happy to answer any questions at the end with that Q&A.
Thank you very much, Molly. A couple of questions. I mean, what an incredible opportunity, I think, first of all, and thanks to Race Against Dementia for providing this funding. A couple of questions immediately come to mind. So it's obviously it can be a subtle but important difference about that you really want the seed of the idea to have already built on existing. You're not looking, which is a big difference between this and a normal program grant. So this isn't to extend, it's for those new ideas that have been sparked from existing research.
Yeah, totally. So the funding is really meant to sort of leverage your current momentum and not necessarily to create a whole new program of research from scratch. So if a proposal kind of stands entirely alone, it's probably a better fit for, say, our fellowships or something. But if it's building on an existing trajectory, then and it could accelerate your progress today, then that's what the fund is for. Yeah.
And that going against. And so am I right in thinking then that this is primarily clearly for fundamental science? This isn't qualitative and care or care research?
That's right. Yeah, it's not for care research. Race Against Dementia, we only fund research into looking into treatments and preventions for dementia. Yeah.
Would that include, say, biomarker research as well, or prevention? Possibly?
Yes, totally. Biomarker research and prevention is 100% applicable, and in fact, that's what Aitana does. So yes, those two would be applicable.
Wonderful. And what else did I have? So what can you? I know I'm jumping. We're going to have Q&A later, but what will and can and can't you use the funding for within this? So can you cover, for example, salaries or lab? You mentioned that you don't pay overheads as part of this.
That's right, but you can use it for salary if you want to. And a lot of our past awardees have hired a research assistant or technician to work on this project, because it's sort of, you have your own line of research and your main program, and this is very much going parallel. So if you don't necessarily have the time to work on the project yourself, you can use this funding to not only for consumables but also for a research technician to do that project.
So you can use it, yeah, you can hire extra people, cover some of your time. You might have to check that back with your institutions as well, because when overhead isn't involved, I know it can be slightly more complicated to use some of that funding for salary costs. But Aitana, I think we might be able to talk to answer some of those questions. Thank you very much. So David Koss, as we heard at the start, is somebody who was awarded funding from this Ignition funding last year, and he's going to talk about his and his lab's research and talk to some of that process. So over to you, Dave.
Okay, thank you very much, Adam. So when I was first asked to give some information from Molly about my project, I really wondered what would be most useful. And I think probably what I want to do is kind of show you at least some maybe slightly modified slides of actually what I used for the interview process. What I would say is that we had a quick run through of this before, and unfortunately on this platform the animations aren't going to quite work, but we'll kind of muddle through. So anyway, I'm David Koss. I'm a kind of relatively early research lecturer at the University of Dundee, and primarily my lab focuses on DNA damage in the context of neurodegeneration. And when I first saw this call, I thought it would be an ideal opportunity to further investigate a potential lead that we had on, which is that there could be a potential of nuclear cytoplasmic DNA being a driver both of Lewy body formation as well as neuroinflammation. So really to kind of pitch this, what is it that we know? Well, we're very interested in dementia with Lewy bodies. This is characterized by aggregates of alpha-synuclein, primarily in the cortex when dementia symptoms are present. Lewy bodies themselves actually occur not only in dementia with Lewy bodies, but also in Parkinson's disease dementia, of course, Parkinson's disease, and they can also be a comorbid pathology present in about 40% of Alzheimer's patients. And so that's really just to say that Lewy bodies themselves and understanding the processes of how they form is a common concern across dementias. We don't really know why these Lewy bodies form, but what we do know is that we know that there's nuclear and mitochondrial deficits that occur early alongside neuroinflammation. And really in the kind of literature at the moment, there's much excitement about the fact that mitochondrial damage can trigger the release of mitochondrial DNA into the cytoplasm. And this can then act via a neuroinflammation pathway, STING-C gas, to upregulate neuroinflammation. And many labs are actually investigating this. At the same time, we know that, at least in vitro assays, that DNA and histones, when in the presence of excessive alpha-synuclein, can cause alpha-synuclein to aggregate. And that's really just to suggest that there could be pools of cytoplasmic DNA which interact with key features of Lewy body pathology. So the work that my lab does primarily focuses on characterizing different types of DNA damage that occur in neurodegeneration. And so we'd already demonstrated that we have an accumulation of DNA damage in dementia with Lewy body cortical tissue. And interestingly, we found that actually this DNA damage is recapitulated at an early pre-symptomatic age in our mouse model. And so this is kind of highlighting the fact that DNA damage could be a kind of druggable target. But what was really interesting was that although it was known that Lewy bodies themselves contain DNA, and this was often thought to be of a mitochondrial origin, we actually began to find enrichment of markers of genomic damaged DNA within Lewy bodies. We saw this in multiple markers. And this really suggested that actually perhaps this genomic DNA damage is causing the leak of this DNA into the cytoplasm. Perhaps that this could be playing a role alongside mitochondrial DNA. And so actually we published some of our findings recently this year. And if you're interested in that, I would encourage you to go and look at this. So what we didn't know is, okay, we have this DNA damage that's occurring in the mitochondria and also in the nucleus, and this is generating cytoplasmic DNA. But we don't really know whether these species or where they're coming from, and indeed if they're really capable of driving alpha-synuclein aggregation or neuroinflammation. So we developed a project, three aims, to look at really to track, firstly, where the DNA damage is first originating in our mouse model. So is that mitochondrial or DNA? And we hope to do that in pre-symptomatic stages. We're also going to use post-mortem human tissue. We've developed a protocol for actually isolating out cytoplasmic DNA in fractions that are free of mitochondria in the nucleus. And in line with Edinburgh Genomics, we hope to have this sequenced so that we can track and determine the relative contribution of mitochondrial and nuclear sources for generating the cytoplasmic DNA. Finally, as proof of principle, we want to take both post-mortem isolated material as well as recombinant DNA or cytoplasmic DNA surrogates and test their ability to drive alpha-synuclein aggregation in a cell model, but also see whether they can enrich pathology in organotypic cultures. So I think it's always important to highlight why your work is important. And foremost, this is really just to kind of provide proof of principle that cytoplasmic DNA may be involved in Lewy body pathology. But moreover, then delineating the timeline of DNA damage in various organelles will give time points for organelle-specific interventions. By establishing in real, genuine cases of dementia with Lewy bodies the source of the cytoplasmic DNA, we can highlight those most pertinent organelles to target and limit cytoplasmic DNA production. And finally, by obtaining this money and developing these models, we'll develop a platform for drug discovery and for future work. And lastly, I just wanted to say about why I applied and what my experiences was. Well, why did I apply? Well, I suppose this was building on a very surprising finding. We didn't expect to see this genomic DNA to be integrated into Lewy bodies. And although this is kind of complementary to our core research themes of looking at genomic DNA damage, it's certainly a little bit divergent. And I guess with that, although it's an interesting offshoot, because we don't perhaps have such development behind the ideas, it might be difficult to find funding elsewhere. I also thought that actually, in terms of the application process, it was very short, and I thought it was a really good exercise in being brief and to the point, and an interesting challenge just for myself to draft that. Now, I would say that I think when I looked through the application I submitted, and I don't know whether it's changed this year, but it was 7,200 character limit, and that includes spaces. And I wanted to highlight that because I drafted mine in a Word document and then came to insert it into the PDF format, and only then did I discover that this character limit includes spaces. So you have to be really, really succinct with what you're saying. And also, I guess because it's a short application, I think people have a thought that, oh, actually, this won't take me too long. It will take you a substantially long time to get the application just right. And in terms of the interview process, my interview was 25 minutes. It was a 5-minute presentation. And the panel primarily consisted of researchers, but also some enterprise entrepreneurs. But most of the questions, I think, were science-based, and most of them were very encouraging and supportive of the idea. There was, of course, a little bit of pushback on my theories and ideas, but overall, I would say it was very enjoyable. And then finally, the last point about this is that there's a really rapid outcome. So the deadline for the round that I applied for was the 16th September, and I knew the outcome by the 28th of November. So that's just a little bit over 2 months, which is really remarkable for obtaining funding. And I guess as a last thought, I think I was really encouraged and impressed with the Race Against Dementia team, and was very excited about the possibility of being able to network and kind of join them for their endeavors. So thank you very much. I hope that's been of some use. And if there's anything that I can answer, you can either ask me at the end of the session, or you can find me online and feel free to email me. Thanks.
Thank you very much, Dave. I'm interested. How have you put the funding to use?
Yeah. Okay. So I mean, we actually, what we did was we decided to hire a postdoctoral researcher. So we're starting to move the project forward now, and of course, we've allocated the salary for the postdoc researcher for 1 year, and also hired, or sorry, not hired, but have paid for the various consumables for the project. And some of this work is planned with kind of collaborators as well, and so we'll provide the materials and resources for them to do that.
And are your collaborators all in the same institution, or elsewhere in the country, elsewhere in the world?
Elsewhere in the world, actually. So we're working closely with some researchers, Fiona LeBeau in Newcastle, but also Tiago Otero in Germany, who has these cell models. And so the idea is that we'll ship this material once we have it over and out to them, so that they can test some of these systems.
Was that detailed in your? Did you have to put that into your application? Is there a place to kind of talk about? Because we know that there's always this tendency with applications that you feel it's going to be strengthened if you highlight the collaborative nature of the research and how many different other people are involved. Did you include that?
Yeah, I mean, primarily it was just in a kind of brackets forms, because you really don't have that much space, right? But you can, knowing that these cell models for aggregation already exist and they've already been published on, it was a case of saying that this is what we would do, using the references and saying to be conducted by, and just having that kind of brief statement in.
Just last question before we come to Aitana,
Sure.
who's very patiently waiting. Did you do the interview on your own? Or did you have anybody else involved?
No. So I mean, I applied as lead applicant, and these are like my collaborators are just that, my collaborators. Yeah.
Brilliant. Thank you very much. Well, let's bring Aitana in now. Thank you very much, Dave. We'll bring in Aitana there. Thank you very much, Aitana. We're going to give you a chance now. We'll go to your slides and we'll hear your story from your application. Let me bring those up here.
Yes.
And over to you.
Hello, everyone. I'm Aitana Sogorb Esteve. This is the story about my application, but also my story with Race Against Dementia. Because I'm a particular case because I was awarded the Race Against Dementia Alzheimer Research UK Fellowship, and I started in 2022 with my project. So this is a 5-year funding. It's different from the Ignition Fund. So I become a RAD fellow and became part of the family of Race Against Dementia. And I'm gonna kind of tell you a little bit about what was this project, what is the project of my fellowship, because it's actually very important for my application to the RAD Ignition Fund. And the focus of my research is synaptic dysfunction in frontotemporal dementia, and I work in fluid biomarkers. And actually, I adapted these slides from one of the posters that we have to do every year for the summit that Race Against Dementia organized. And Molly showed before a picture of us last September, was it? Or July, I don't remember, but at Aston Martin factory. So we gather all the fellows and RAD associates and the Ignition Fund awardees for a few days, and then we share our research and our experiences. So in this poster, I kind of presented my fellowship and also a little bit of the RAD Ignition Fund. So it has a lot of text just because it's an adaptation of that. So I hope you can see it properly in terms of the figures and so on. But yeah, so my fellowship focuses on exploring and identifying synaptic dysfunction in frontotemporal dementia through fluid biomarkers, because sadly, we still don't have good fluid biomarkers for FTD, as a different of Alzheimer's disease, in which we have gold standard fluid biomarkers that help with the diagnosis and monitoring, therapeutic interventions and clinical trials and progression of the disease. So for my study, I usually, but I use proteomics to study proteins that are related to synaptic dysfunction, but other processes in spinal fluid samples or blood samples from a specific FTD cohort, which is the Genetic FTD Initiative, or GENFI. So I'm focusing on familial frontotemporal dementia, because using these type of samples and these participants, at least I know the genetic status of them, and then it will be a little bit easier for me to interpret what I'm finding in the CSF or blood and related to the underlying pathology. So this is just a summary of the first part of my fellowship that resulted in a paper, proteomics paper, that we published in February this year. Basically, what we found was specific protein profiles for the different types of genetic FTD. And this figure that you can see here is just a summary of the pathways. We did a weighted gene network co-expression analysis of our proteomic signature, and then we saw different processes or pathways that might be altered in our sample. And basically, we saw core biomarkers that we know that they are altered. We saw synapse models, that is actually the focus of my fellowship, but then we saw something that was a bit unexpected in our population. And this is what is actually the basis of my Rat Ignition Fund. So if you don't know a little bit about frontotemporal dementia, there are 3 main genes causing for FTD: expansions on the C9 operating frame 72 gene, mutations on progranulin, which is a protein that is involved in the endolysosomal system, and also mutations in MAPT, which is a protein codified in tau. So in FTD, we have 2 types of pathology. Mainly, there are many more, but the main ones is a primary tauopathy or a TDP43 proteinopathy. So because of the role of specifically progranulin in the endolysosomal system, we thought that we were going to see changes in this lysosome model or cluster and these proteins that are related to the endolysosomal system in progranulin mutation carriers. However, what we found was that this model or this cluster of proteins was decreased in symptomatic MAPT mutation carriers, and actually 2 of the proteins in this cluster, DNase2 and PLBD2, they were very good differentiating MAPT mutation carriers from either C9 or progranulin. So this was something that was really unexpected. We didn't thought that that was going to happen, and we thought that it was really, really interesting, and no one has reported that before. So that was actually the idea that I had to, okay, I think this is very fit for a Rat Ignition Fund, because there's something that is not on my the focus of my fellowship, because I'm focusing on synaptic markers, but it's something that is really worth to explore. So yeah, so what I took this result, I also included this figure as a preliminary data supporting my application, and what the project was to explore. We've seen this change in CSF because this project was done in CSF, but then can we measure these changes in blood? So my approach, the same one as my fellowship, is to use extracellular vesicles extracted from plasma, but the focus of my main funding pod, which is the fellowship, is to study synaptic dysfunction. However, this technology of using extracellular vesicles can be applied to other things. So my idea was to also study in these extracellular vesicles endolysosomal proteins. So yeah, that was basically the summary of my project and how I came to the idea and how I came to the applications. And I think the key point is that the results that I got from the first experiments of my fellowship were very unexpected. And the results were also innovative. We haven't seen publications exploring that or seeing similar results in MAPT mutation carriers, also because FTD is rare form of dementia, and there are not that many genetic cohorts in the world. So it was like very specific and innovative. And also the funding, I think it's very flexible. They are looking for innovative ideas. And this has been discussed before, but what I needed at that time in my project, I got my salary covered from my fellowship, and I got a big pot of consumables for my fellowship, but I actually needed more, like I needed help in the lab, basically. So I allocated the Ignition Fund to hire a research assistant for, I think it was 22 months, according to the UCL salary. And this is what really made a difference on kind of pushing the project forward, because having help focusing on this specific bit, then it gives me more time to focus on my main fellowship, and it has made a big difference on that. So yeah, I'm very happy to take questions about this, and thank you so much.
Thanks, Aitana. So let's bring everybody back now. Here we are. There's Dave again, and there's Molly. Hi, everybody. Thank you very much, Aitana, for over. I should give you the same question. Although I think you actually answered how you allocated the funds already. You already answered that.
Yes, exactly.
Let's jump. We have had some questions in. Let's see. We're going to put Molly to the test now and see if you can take these questions, Molly. So the first question is, would you include digital biomarkers within the scope?
Absolutely, yes, I think we would. Definitely. I mean, like it's about, if we're trying to work towards ways of treating and improving our understanding of dementia, and if that is what you'll be able to do in your project, then absolutely, it's definitely applicable. So that sounds very interesting. Yeah.
I guess digital biomarkers is things like digital cognitive testing, or things that have come from smart devices. We know the DRI, which is where I think you're in the DRI, aren't you, Aitana? There's a whole institute there dedicated to technology. So that sounds like a positive. Next one we have is, I wonder if this grant is focused more on wet lab work, or could it extend to neuroimaging data? Example, vascular imaging markers, atrophy markers, or previously collected markers.
Again, yes. Sorry, I've lost you guys. Can you still hear me?
We can, yeah.
Okay, cool. Sorry. No, yes, absolutely. Yeah, I think most of our previous applicants have been more wet lab based, but we would definitely welcome applications from neuroimaging data, and again, still really important questions that you're going to be asking there. So yes, absolutely.
And our third biomarker question, probably, sorry, my fault for suggesting it comes from Amy, who asks, can the funding be used to generate biomarker data in new sample? I guess this, I'm guessing you're going to say this depends on whether it's still building on.
Totally, yeah. If the new sample is still something that's going to complement your existing work, then absolutely. But again, if it's maybe going more into the territory of starting a whole different new program of research, then perhaps not. But I think, yeah, that really depends how it kind of sits along in the work that you're currently doing. Yeah.
One of our previous questions, they've just replied to confirm that it is about psychological sensors in the home, and they thank you for your presentation. So yeah, that makes absolute sense. So do keep your questions coming. We'll make sure we get to those. Let me go down my list now and ask Aitana and Dave, what was the most challenging part of the application process, apart from trying to, sounds like really trying to bring that down to the character limit? Is this a small, I mean, Molly mentioned that this is a tighter application than what you might usually apply. Is this also tighter on things like character limits? Do you have to have a skill to really drill that down, would you say? Dave, you mentioned character problems.
Yeah. I mean, I think, yeah, I mean, it definitely was a challenge. And I think, the number of drafts and redrafts and the number of swear words I stated when I realized that it was also the spaces were counted. And I thought, actually, out of everything that I've said today, probably mentioning that the character limit includes spaces is probably the most useful thing for everyone, right?
It's definitely a top tip. What about you, Aitana? What did was there anything that gave you pause for thought before you decided to apply, or was there,
Well, I think it's, I really like this type of applications because it makes you work a lot on getting to the point of what is really important. And it's, I think it's because, when you apply for projects and fellowships, they are very worthy. And this is just like, okay, but what is really important to what I'm doing, what I want to study? So it helps you to focus a lot. So yeah, I think I really like that. And that was actually the most difficult part of it.
So, so this is probably an important point. There is it's not so much about also what you include; it's what you don't include. That sounds like the important, because I know that we all, we've all been to those conferences or started an application where you start by using those first 500 characters just to explain what the disease is. I mean, where do you sit on that, Molly? How much of that kind of background do you really want to set the scene, or would you rather them skip it altogether?
Yeah, so there is the first part of the application that I think the subtitle is actually background, And I think now what we're really trying to understand, just build a picture of the specific research landscape there, and just where the gaps are and how you're going to fill it. We don't necessarily need a whole story, and the people on our panel will have a general, like, they're all dementia professors, so they might not know your specific area of research, but they'll have a very good understanding and background themselves. So yeah, we don't need lots and extensive detail, but like Aitana said, just get to what's the really important part, and that's what we'll need to know.
And was there any, Aitana and Dave, what kind of support or feedback did you draw upon when preparing your bid? I think you mentioned drafts and redrafts. Did you get, have you involved the people and your collaborators to look at this?
Sure. Do you want to go first, or?
No, you can go first, Dave. Yeah.
Okay, thank you. So I think what I did was I had several practice interviews, actually, because I think that was, again, the other challenging part of this is that you had a 5-minute kind of presentation as well, and again, that's you've maybe done the hard work when you've written the application, which is to kind of cut out anything that's kind of superfluous, but you still have to get across your message and your vision in those 5 minutes. And it's probably the same for all interviews. I always want my worst interview to be the practice, right? Mm-hmm. So, and I make sure that I ask not only, those colleagues that I count as friends, those that I know are really kind of very critical and very demanding, because that's, that's the way I'm going to learn how to defend it, right? So, yeah.
And you've just given me an idea now that we could probably, between us, we could set up a little mock interview thing, couldn't we? That if anybody who's applying who gets through to the interview stage would like a mock interview, drop me a line and I'll happily drag some people together to put you through the ringer. No, not really. To do that mock interview. I'm sure I can draw upon some of the previous applicants and things as well to help you with that. So do drop me a line if you get through to that stage. I know you're not going to know for a while, and we'll see if we can help. What about you, Aitana? Was there, how did you prepare?
Yeah, so I think it's because it's a short application, it's easy for collaborators and supervisors to review it because it won't take them that much time. So I make sure I ask a lot of people to kind of review it. And is it clear? Do you think I stress enough the importance of it, on how unexpected it was from my project? Is it separate enough from my project? So yeah, I ask a lot of feedback on the written form as well.
I'm just trying to go through my questions now, which are more for Molly. What does a strong? No, I think you've kind of already answered that. Are you expecting any preliminary data in this, or is it acceptable to propose something entirely speculative?
Yeah, so you don't need any preliminary data to apply for the Ignition Fund, and certainly, you don't need that data published at all. And that, yeah, both Dave and Aitana didn't before they applied. But if you can, you just need to be able to really explain exactly what it is that the finding that you do have that is unexpected, and you don't need lots and lots of detail. It can be very speculative because we also know that maybe you might go down this line of inquiry and it might not work out. But that's okay. We just want to give the opportunity to be able to go down and really investigate these ideas that you have. So you don't need specific preliminary data. We won't ask to see it, but if you can give an idea of things that you have found, and that's kind of really what it's for. Yeah. And I also, sorry, going back to the interviews, I thought I should also mention that we sort of gave applicants also I think it was 3 or 4 questions that we were going to ask during the interview that the panel had the questions that when we were discussing their applications, the panel had sort of had questions about, and so we gave those to the applicants before the interview so they could sort of prepare some answers too, because I think the way that we wanted to sort of shorten the application process, I know often you sort of can do a bit of a rebuttal, and we can give you the feedback, you reply to the feedback, and it kind of goes on like that. And to keep that shorter, we just thought we would do that in the interview, so it becomes a bit of a live rebuttal as well. So that's also one way of it.
Were there lay people on the interview panel as well?
Not on the interview panel, no. The lay reviewers only review the applications at the paper application stage, but their feedback and their thoughts are brought into the initial scientific panel, and then also in the interview as well, especially when we're trying to sort of really distinguish between the top applicants, and the lay reviewers can give really specific and personal feedback about the impact of the projects, and they're really helpful to do that.
Is there anything you do? So I'm just thinking back to your fellowships, which of course closed a few months ago, that in that you were particularly keen to see a machine learning or an AI element to that particular research. Do you have any particular areas that you'd really like to see this time? Are there any gaps, or is it really just open? You're not focusing at all?
Yeah, we don't have a particular focus really for this particular call, but like you said, we're always really keen to see ideas that we haven't seen before, and particularly with artificial intelligence and machine learning, and perhaps even bringing people not necessarily dementia researchers, but maybe people with more of a background in data science or artificial intelligence, bringing that expertise within dementia research as well. So yeah, I mean, it's open to all UK early to mid-career researchers, no matter if you're in dementia or not. As long as your project is about dementia, then that's what makes you eligible.
So good to think creatively then, not just on the new ideas, about the kind of collaborators you bring in. We mentioned before that you have to be funded via a UK institution for this, but collaborators and others can be literally anywhere in the world, but it's contracted through a UK institution. We've had a good question, which I was going to ask myself actually, and this has come up from the audience, which is, how many proposals are you likely to fund, and are you assuming that everybody will apply for the kind of £100,000 that's available?
So it will really depend on the amount and the quality of the applications that we get. We first ran this call in 2023, and we funded 3 applications from that, and then last year we also funded 3. So I think it will probably be up to 3 this year, but perhaps more if we really get a high quality amount. And not everyone does actually apply for the 400. We had a few who are slightly below, and that doesn't, that won't affect your application. So yeah, we don't assume that, so yeah.
I can speak to this. I know that Race Against Dementia are one of, kind of incredibly in the past have always, I guess it's because you're this kind of smaller and really dynamic charity, you've been able to respond quite dynamically to the applications that come in. So when you might have in mind that it could be 3, but if you've had more applications, you've been able to say, actually, we'll move money around, we'll find, you can draw on your supporters and things to say this really should, all these studies should be, I mean, clearly you're not going to fund 20, 30, but if you get extras, and that's one of the great things about Race Against Dementia, and that they do. We're nearly out of time. Last chance, anybody, for asking any questions. What are the ways I'm going to round up on this? So anybody who doesn't already come along, for those that don't know, Dementia Researcher has a community app, which is where we have a kind of sport community, and others like you. There's over 1,000 members there now. We hold a weekly webinar every Wednesday at 12 noon or 8 p.m. talking about research and careers topics. And one of the ways we round off all those now is by explaining that Dementia Researcher's website's got over 3,000 pages of blogs, posts, webinars, live streams, podcasts, all of which are transcribed. And then we've trained an AI model to learn just from the content on our website. So it also includes all the things like the jobs and the grant applications and other things, everything we ever put in. It hasn't learned from the internet. It's only learned from the content in our website. And so I asked that model this morning to give me 10 top tips to write a strong ignition fund application. Shall we? I like to round this off. I'm going to give you these 10 top tips now, and we're going to see if you agree and if we've already covered them. So number 1, start with a truly unexpected insight. Anchor your proposal in a surprising and counterintuitive result that challenges assumptions. This is the fund's core purpose. Make it crystal clear. Absolutely. Right. Start. First up, this is a counterintuitive result. It is new. Sentence number 1. And number 2, keep it focused and concise with strict character limits. Every word matters. Aim for clarity, relevance, and directness in each section. Edit ruthlessly. I think you both all said that. Number 3, separate this from your existing work. Explain how your idea couldn't be pursued with current funding, highlighting why this is distinct, disruptive, and previously unexplored. Because that's a good question. If you're trying to kind of just get extra money to build on your existing grant for something you're probably going to do anyway, this isn't the grant, is it? This is something that otherwise wouldn't get done because your existing grant doesn't cover this. So make that clear. I think is that also true? Everybody's nodding. Number 4, design a fast, feasible experiment. The fund supports projects that deliver insights quickly, ideally in 12 to 24 months. Choose a plan that's practical but can also produce meaningful findings. Reviewers always pick up on this, don't they? Over-ambition in grants, that you're trying to do something that's really going to take years to do. You've got 12, 18, 24 months to do this. So make sure what you're saying can be done in that time. Use your budget wisely. Only include costs for people and materials. No lab space, no overheads. Be specific and transparent about what the money will be used for and why it's essential. Number 6, write a truly accessible lay summary. The lay summary panel of a personal experience of dementia, but no specific background. So avoid jargon and explain project's real-world value clearly. And number 7, tailor your lay project title and aims. Your lay title must be short, engaging, and entirely free of technical language. Make it easy for anyone to understand. It's a bit of duplication from the last one, I feel there. Number 8, show potential for real-world impact. In both scientific and lay sections, explain how your findings could influence the future of dementia research, even if it's at a clear and early stage. I don't know, would you agree with that? Yeah?
Yeah.
Number 9, demonstrate your passion and readiness. Whether you're an early career researcher, whether you're just finishing up your PhD, a postdoc or senior postdoc, use the biography section to show why you're well placed to take this idea forward. And again, bring in those collaborators. And last but not least, number 10, get feedback before submitting. Ask a colleague to review both scientific and lay section. A pair of fresh eyes can catch unclear phrasing or unintended jargon. Ta-da!
Excellent advice.
AI's pretty good, right? So I'm going to flag, we last chance, we didn't get any further questions, but I'm just going to flag up a little ticker along the bottom of the screen just to remind you all that the application form is a PDF file which can be downloaded from the Ignition Fund website, which is raceagainstdementia.com/research/rad-ignition-fund. There's a QR code going to appear any second now at the top of your screen. There it is. There's a QR code there, which you can take a screen grab on your phone. I'll grab your phone now and click that Ignition Fund. You'll also find a link on the Dementia Researcher website, or if you're watching this on YouTube or on X or LinkedIn, it's in the event description as well to get you straight to that. The deadline for applications is 4 p.m. on the 12th of September. If for some reason that changes, keep an eye on the Dementia Researcher website and we'll update that. But at the moment, that's the deadline. That should hopefully give you all of a chance to get your finance departments and others to look at that beforehand. I know we're coming into the summer, but don't wait till the last minute to do this. If you need to start now, get it into finance departments in July, because everybody's going to go on holiday as soon as the school holidays start, right? But thank you, Molly, for giving everybody a little bit of time into September for that final bit of polish that they might want to add to that. And again, once your application forms are completed, or if you've watching this on catch up and you've got questions or you've thought of something that didn't get asked, you can email ignitionfund@raceagainstdementia.com, when Molly will take your questions. Thank you very much. I'm just seeing if there's any final question that I missed out on. Oh, my question didn't get answered at the recap. I think it did, Adriana. So historically 3, but not fixed, and you can apply up to 100,000. Not everybody does. It's not going to impact you if you don't apply for the full grant amount. There's been 3 before, but fundamentally, if there's one good funded one, or possibly 5, who knows, that they'll flex the grant according to the application quality of the applications that come in. Any final takeaways? Any last tip you'd give for an advisors, Molly? One, I'll give you a chance to have a final thought for people.
I think just be focused and clear, and really show us what unexpected finding you have.
Brilliant. What about you, Dave?
I think if you feel like you have a good idea and that you're willing to apply, I think you should go ahead and get working on that, and don't be put off by the kind of slightly unusual application process, because I think you'll learn a lot of good skills along the way to do that.
Aitana?
Yeah.
You get the final word.
Well, I agree with Molly and Dave, actually. Just put some thought on how to structure it in the way that is the most focused and clear application you have ever written, because that will pay off. Yeah.
Thank you very much. And thank you very much, of course, to Race Against Dementia. We're very proud to be funded by ourselves at Dementia Researcher alongside other funders, but Race Against Dementia's generous contribution towards our costs really make a massive difference to ensure that we can keep going to support early career researchers and, of course, the wider community as well. And, of course, happy birthday to Sir Jackie, who I believe it was Sir Jackie's birthday and vision for all this that's made it possible. So thank you very much. And if you're watching this and you're not a researcher and you'd like to contribute, I know Race Against Dementia are always willing to and looking for donations. You can donate to the charity at their website, and they've got a great little shop as well where you can buy merch. So have a look at that if you'd like to donate very generously to Race Against Dementia. Thank you very much, everybody. We'll round out with that wonderful video again, and thank you all very much, and we'll see you again.
I've had a lot of challenges in my life. We went through some tough times. We lost a lot of friends. And Helen was always there, supporting me through everything. She was razor sharp, brilliant with that stopwatch. Today, we're facing a new challenge. Helen has been diagnosed with a terrible disease. Dementia is nothing less than a worldwide epidemic.
Indeed, 1 in 3 people born today will develop some form of dementia during their lifetime unless we can do something to change that. And whilst there has been important research progress, it clearly is not fast enough.
When I was racing, a pit stop might have taken 2 minutes or more. These days, it can be under 2 seconds. Formula One moves fast. The pressure to perform is relentless. High technology problem solving is what we do every single day. By contrast, medical research can be painfully slow. We bring Formula One technology into the laboratories. We've identified the best research talent in the world to work with the leading Formula One teams for a period of time to learn from their performance-driven culture and to adopt their sense of urgency.
I developed drugs from scratch, and I followed the Formula One mindset to treat dementia. So we need to find new ways of doing things smarter if we're going to beat this really cool disease.
So I'm hoping with my research to find something that has a tangible benefit, drive towards a real change, which can be translated into clinic, that can make a difference to patients, because that's what we really need.
This is not a race that one person is going to win. Science builds on little puzzle pieces that get put together. There has to be a team effort.
I'm working to beat dementia faster.
I'm working to beat dementia faster.
I'm working to beat dementia faster.
Let's beat dementia together.